Welcome, everyone. My name is Renee Duprell, and I'm the Director of Development here at the Institute for Systems Biology. And I apologize, my camera is not working. We're having a bit of a technical difficulty. So the good news is that our speaker's camera is working, so you will get to see Nitin as he speaks. Thank you all for joining us for our second research roundtable of 2022. This is our virtual series that we host to keep you up to date on research at ISB. This series is geared towards non-scientists, and our goal is to make our work more approachable for all. Our intention is to have one of these just about every month through the end of the year, so keep an eye out for our emails. Today, we'll be hearing from our featured scientist. Then there will be an opportunity for questions and answers after that. There's a Q&A box at the bottom of your screen, and you're welcome to submit questions at any time. I'll be monitoring those, and when Nitin is done with his presentation, we'll move to the Q&A session and we'll work through those questions. Our speaker today is Nitin Baliga. Dr. Baliga serves on the leadership team here at ISB. He is a senior vice president, director, and professor here. Nitin leads a cross-disciplinary team of scientists to address complex problems relevant to global health, personalized medicine, energy, and the environment. Dr. Baliga's work has been published in top international journals, including Cell, Science, and Nature. In his spare time, Nitin makes transformative contributions to science education. Science curriculum developed by Nitin's programs has impacted over 2 million students in over 100 countries. That's a topic for another research roundtable, and I promise we will cover that. Welcome, Nitin.
Thank you, Renee. Good afternoon, everyone. Let me share my screen. It's great to see all of you. There you go. So I know these are unusual times, and I think we're going through what might be once in a lifetime experience for pretty much everyone, going through a pandemic of this magnitude and being locked down for such long periods of time. But pandemics are ongoing around the world and have been going on for decades, and tuberculosis pandemic is one of them. It's just that we in this country have not experienced it, and it's out of sight, out of mind. But this disease has laid waste to many communities, and I'll tell you a little bit about the disease and then tell you what some of the challenges are going forward and how we can address them. So if you don't know what tuberculosis is, this is a very quick primer. So it's a disease caused by a pathogen called Mycobacterium tuberculosis. It's a bacterium. Even a few cells of the bacterium transmitted through aerosols can cause the disease. In most individuals, our immune systems will naturally clear the infection, and that can be through the innate immune system or by our acquired or adaptive immune system. But in some individuals, the infection can proceed and go into what is called a latent TB infection, and it can form the structure called a granuloma. I'll tell you more about that in a little bit, and it can stay in that state for years and decades. And over the lifetime, latently infected individual has a 10% chance of getting reactivated and getting full-blown disease. Now, in the early stages when that happens, the bacteria become metabolically active, and there's now a proposal that we should have one more stage of TB called incipient TB, where the bacteria are metabolically active, although you may not see any abnormalities on lung radiology. But thereafter, you see abnormalities in the lung through radiology, but you don't have symptoms, and that is called subclinical TB disease. And then when you have full-blown disease, you can see all the symptoms, and in late stages, it becomes very severe, where individuals are breathless, they are constantly coughing, there's high fever, and eventually that leads to a very painful death. And here are pictures of some individuals with tuberculosis. And if anyone's ever been to a late stage TB ward, you can see the extent of suffering that the patients undergo before they die. So it's a terrible disease. Now, there's a large number of people around the world who are latently infected with TB. In fact, a third of the world's population, about 2 billion people, are estimated to have a latent form of TB infection. Of those, as I mentioned earlier, 10% go on to get the disease. So on average, every year, about 10 to 11 million people get active tuberculosis. And of those, about 1.5 to 1.8 million people die from TB. So that's about a death every 20 seconds. So this is now only second to COVID in the last year as the leading killer infectious disease. Now, there are cures, and for people who get TB, they will be put on a 4-drug regimen for 2 months, and the drugs are isoniazid, rifampicin, ethambutol, and pyrazinamide. And subsequently, they get 4 months of double therapy, a maintenance therapy with isoniazid and rifampicin. And overall, the 6-month treatment will clear the infection in most individuals, about 95% of the cases. But 5% of the people who get treatment will relapse. If you don't get the treatment, about 50% of the people who get the disease die from the disease. So the treatment is very effective if the infection is by a sensitive strain of TB that is sensitive to all these drugs. If you get an infection from a resistant strain, then there's different kinds of resistance. There's multi-drug resistant strains, which are resistant to isoniazid and rifampicin. And then you have extremely drug resistant strains that are resistant to isoniazid and rifampicin, and another drug called fluoroquinolones, and one of 3 injectable drugs, that's kanamycin, capreomycin, or amikacin. And these strains are very difficult to treat. There are other strains called totally drug-resistant strains that don't respond to any treatment. But even getting rid of a multi-drug resistant strain is extremely difficult. And the patients end up taking 8 to 10 pills per day, or 8 to 10 drugs, I should say, and multiple pills per day. And these are not trivial drugs. They come with a lot of severe side effects. And so these are some of the side effects: hearing loss, depression, or psychosis. In fact, there's a higher suicide rate in patients with MDR TB or on this drug regimen. Kidney impairment, there's peripheral neuropathy. And so there's a desperate need to find drugs that are more effective. And I'll get to some of the criteria that are considered important for new TB drugs in a minute. Now, those are the number of drugs I showed you in the previous slide, are the number of drugs you take per day. That treatment can continue for 2 years or up to 30 months. And so over that course of treatment, a patient may end up taking up to 15,000 pills, okay, with all the side effects I described. So this is a very serious issue if someone gets multi-drug resistant TB. And even after getting that treatment, only about half of the patients are cured. So and that's partly because the drugs, 4 of the drugs that I used were developed for TB. The others that I used were not specifically developed for TB. They are repurposed from other uses. And I just want to make a side note here that drug resistance, or antimicrobial resistance, AMR for short, is a growing threat, not just for TB, but for multiple diseases. There's AMR strains that are cropping up for all kinds of diseases. And the estimates are that by 2050, about 10 million people will die because of infections associated with drug-resistant pathogens. And of those, even just now, more than a quarter of the fatalities are because of tuberculosis. And I just heard from one of my colleagues in my lab that just in the last year, there were about 2 million deaths that were attributed to AMR-related infections. So this projection seems to be not far-fetched. So what do we need to do? We need to find new drugs that are more effective. And there's a bunch of criteria that have been identified by the World Health Organization and experts in TB. And here are 5 of the things that they've identified. One is we need drugs that are effective in killing TB in a short period of time. So you don't have long treatment regimen for months and years. We need treatments that can target not just the susceptible strains, but also these multi-drug resistant and extremely drug resistant strains. We also need to simplify the treatment, meaning instead of having many drugs, having few drugs that are taken in few pills, because compliance is a big issue. If patients stop taking medications because they're too complicated or there are too many side effects, then they have the risk of one, not being treated fully and not being cured, and two, of gaining drug resistance and spreading it. We also need lower dosing frequency. So some of these drugs can be cleared by our metabolism very quickly. So we want drugs to stick in the system for a longer period of time, which also means they should not be toxic. Okay. And finally, a lot of these reactivations that happen in TB when people go from having latent infection to active disease, it happens because of their immune system being compromised. And one of the ways it can get compromised is by HIV infections. And the drugs used to treat HIV can interact negatively with drugs used to treat TB. So we need drugs that do not have negative interactions with HIV medications. So to be able to get these 5 different criteria met, there's a bunch of challenges we have to address. And I'll go through those and give you a summary of why those challenges are important to address. So number 1, and I'll show you in a little bit how TB can exist in a very heterogeneous set of states, phenotypes, inside a structure called the granuloma, which I mentioned earlier, but I'll touch on more in the coming slides. And in the granuloma, because they can adopt these heterogeneous states, they can resist treatment by multiple drugs. Each of these states have different susceptibility profiles, meaning they can be killed with some drugs, but not with other drugs. And knowing exactly how many different kinds of states exist and what their drug susceptibility profiles are is important to come up with a treatment regimen or drug cocktail that can kill all of the bacteria. And for the reason I just described, drugs that we discover in the laboratory in flasks, in liquid cultures, don't really address all of the heterogeneous states of TB. And therefore, they are not as effective when they're used in the clinic on a patient. So we need to use animal models, which makes it really complicated because you can use a mouse model that kind of approximates the human disease, but not exactly. And you can go to non-human primates, and they do simulate the human disease, but they're extremely expensive, and there's all kinds of ethical issues, etc., related to that, which makes it very difficult to do very high throughput screening. So you can't screen many molecules, and it's also very, very expensive to run these studies. Okay? In fact, drug discovery, just in general, not for TB, is very expensive, and it can take years, 15 to 20 years to find effective drugs, going through all the different clinical trials. And the cost of finding drugs can go up to $2 to $2.5 billion per drug. And we're talking about finding drugs that will be useful to treat a disease in the developing world where people barely have a few $100s of income, annual income. So we cannot develop drugs that are extremely expensive to use. In fact, a lot of pharmaceutical companies have backed off from developing drugs for diseases like tuberculosis because there's no incentive to develop these drugs. So we need to make the discovery process a lot cheaper. And going to human clinical trials, all of the challenges I described above are relevant, but it's also very difficult to know whether the drugs you're giving to a patient are actually working, because the typical assessment happens 8 weeks after the treatment regimen begins. And this is a slow-growing pathogen. It takes a few weeks to grow, and you have to work with it inside a containment facility called a BSL-3 facility. And it's not easy to do the work in these environments. You have to wear a spacesuit, you have to work inside a hood, etc. So you could find out in 8 weeks that the treatment you've been given a patient is not working at all. So clinical trials essentially are difficult to monitor on a timely basis. And primary reason for that is what I just described is the bacteriologic assays, is growing the organism on plates and then counting the colonies they form, etc. That is very time consuming and not a very efficient approach. And there are other issues as well. In addition to taking a long time, these assays are not accurate. They cannot identify Mycobacterium tuberculosis when they're in a state called non-cultivable or non-culturable state. These are states in which the bacteria can hide and stay dormant. And they don't show up on plates, so you might assume they don't exist, but they do. And I just mentioned about the need for special facilities. So here at ISP, we're working on two big ideas to address all of these challenges and to accelerate TB drug discovery. First is that we want to use what we're known for, which is systems biology. So look at all the genes of the pathogen, its interactions with all the host cells, etc., and do that at a systems level rather than doing it at one gene at a time. That's systems biology, combined with machine learning and artificial intelligence to then mine through all of the data we can generate to find drugs that are more effective and affordable. That's one idea. The second is we want to find ways of replacing these time-consuming and inaccurate bacteriological assays with more affordable and sensitive molecular signature-based assays that are faster and more sensitive in detecting not just the pathogen, but also whether it is responding to therapy. Okay. The work that I'm going to describe is funded by both National Institutes of Health, NIAID in particular, and the Gates Foundation. And we can't do this alone. This is a big global problem. So we have a large team of collaborators around the world who are helping us with this. And I won't name them all right now, but just point out that they cut across North America, Europe, South Africa, and Asia as well. Also in Vietnam, which I haven't shown here, but I'll point out some of these partners as we go through this presentation. And they fall in different areas of applications from diagnostics, drug development, and vaccine development. So let's revisit the life cycle of TB. And TB and humans have interacted with each other for thousands, if not tens of thousands of years. And this is the life cycle it has gone through over that period of time. So the disease or the bacterium is disseminated through aerosols. So these little droplets, very similar to how COVID is disseminated. And when the bacilli go into an individual, a few bacilli, 1, 2, 3 bacilli are sufficient to cause the disease. In most cases, as I said, it will get cleared, but in many cases, it will go into this latent infection form, which is a contained form, which can get reactivated. In some cases, the infection can directly proceed to active TB. So altogether, in the end, you end up with about 10 million active cases, 10 to 10.5 million. And within an individual, if you zoom in, what happens is the pathogen is first engulfed by these macrophages called alveolar macrophages, because their job is to try and clear these invading bacteria. But this bacterium, because it's interacted for so long with humans, has become really adept, excuse me, at adapting to all the tricks that the macrophage plays to try to kill it. So first thing the macrophage does, starts lowering the pH. So it makes it more acidic inside this phagosome. So it can then merge with the lysosome, which has the enzyme lysozyme that can kill the bacteria. But by the time the pH has dropped, the pathogen has activated networks and programs to change its cell wall. It makes this cell wall called mycolic acids. It's a waxy lipid that can prevent things from getting in, and it prevents the enzyme from getting to the cell wall, which is made of peptidoglycan, so it can protect itself. In fact, it can then take advantage of the immune system. So the macrophage sends out signals and other immune cells start coming in, and they all try to kill the pathogen, but it starts building this nice little house for itself. And in the end, you have different kinds of cells of the host, and there's a periphery of T cells, and you can see the pathogen inside different cell types, okay, inside epithelial cells, you're seeing inside macrophages, alveolar macrophages, interstitial macrophages, and inside all of these cells types, the pathogen experiences different environments. low pH, low oxygen, low nutrients, and it has programs that elicits to deal with all of those different challenges, and that's why it exists in this different heterogeneous states. Okay. This is the stage, the solid granuloma, in which a latent infection can exist for years and decades at a time. But when it goes into reactivation mode, the pathogen becomes metabolically active, it starts dividing, it breaks open the cells, and it comes out into this necrotic granuloma, and they lie outside the cells, and here they may be in a different state as compared to in each of these different cell types. And as they continue to grow, they go to a caseous granuloma, and that's when disease becomes severe. The patient starts coughing severely, and the pathogen is disseminated. It exits the granuloma and then goes into the sputum and aerosol when the patient coughs. So through this life cycle, TB has learned to deal with a variety of different challenges: the immune response, hypoxic environment, reactive oxygen species. These are all the different ways that the host is trying to kill it, but it has learned to develop these programs, these genetic programs to deal with it. You can think of genetic programs like recipes, like you have a recipe book, and depending on context, if it's Thanksgiving, you will just cook with recipes that are relevant for Thanksgiving. You would make turkey and you would make mashed potatoes, etc. So similarly, there's 4,000 genes in TB. Not all of them are acting at the same time. Depending on the context, some subsets of genes will be expressed and they will generate capabilities for the pathogen to deal with the challenge at hand. And we need to know what are these programs and when do they manifest, who are the genes involved with the programs, and in those programs, what are the vulnerabilities. To do that, we developed a series of technologies at ISP. I mentioned some, and these are all the wonderful people who have contributed to developing these technologies. And Vivek, for example, developed this technology called PerSort, which leverages an instrument that can scan thousands of cells per second, but he's figured out a way of marking cells that are existing in that persister-like state, that hidden state. And they exist in very small numbers within a larger population. So he's figured out a way to map these cells. So using that instrument, we can now find these cells and we can sort them and enrich them, and we can break them open and we can read all of their genetic programs, okay, all the genes that are expressed, all the transcripts and so on. We've also developed these reactors. As I mentioned, working in the BSL-3 is very difficult. And some of these experiments we have to do over many days and nights. And we want to study how the pathogen responds to, say, a shift in oxygen or a shift in pH. And we want to capture the sequence of events that are triggered by these changes. And to do that in the laboratory in a flask is very difficult. So we developed these automated reactors. Abrar and Eliza developed this, so you can have these fiber optic cables connected to computers that you can then access remotely. So Abrar would be sitting in at his desk or in his room and constantly monitoring the cultures, and then he would come in and collect the samples at the right time. So we could slow down the transition of the pathogen when it's shifting to responding to a shift in the environmental change, and capture all the events. And Eliza similarly has developed a technology to then enrich the genetic material of the pathogen from within infected host cells, because the pathogen genetic material is in very small amounts compared to the host material, and you cannot really quantify it because of that. But using this technology called PathSeq, we can enrich the pathogen genetic material and sequence it. And with all of these capabilities, we can now apply a systems biology approach to then reverse engineer the network. So I should point out, we are not the only people doing these studies. A variety of different labs have done these studies and are doing these studies. So we've compiled data from all of those labs. And these studies have essentially investigated how different perturbations that the pathogen experiences influence the expression of genes. And these conditions here simulate what the pathogen experiences within the granuloma. And we relate it to different outputs, different phenotypes, ability to grow in macrophages or go into dormant states and so on. And then we can collect these genome-wide measurements of how genes are expressed and take that information to do analysis with machine learning and other types of network inference algorithms that give us the regulatory circuits that are controlling which genes are expressed and in what contexts. We can then integrate the regulatory circuit or regulatory network with how those changes would influence metabolic reactions across the entire physiology of the pathogen. And in that, through that series of steps, we build this predictive models that now allow us to predict what would happen if we were to perturb the organism, the pathogen in certain ways, and exactly what networks are responsible in what context. So I've shown pictures of all the people who have contributed to this work. And I'll just jump to, instead of showing you more of how the sausage is made, I'll show you exactly what type of information we can learn. So these are the models that we've developed. They've been published, and I won't spend time on that anymore. So here's an example of a study we did several years ago. And Eliza and Shui, who was then in David Sherman's lab and now is a faculty member at Seattle Children's, they did this work. What they found was when the pathogen is treated with a drug called bedaquiline. It's one of the new drugs that has been approved for treating TB and multi-drug resistant TB. But the pathogen has learned through its evolutionary experiences ways to deal with this new drug. And what Eliza and Shui did was they applied the systems biology approach, and they figured out that when you add bedaquiline, a very specific network is activated in the pathogen. And that network can then trigger all kinds of capabilities that give it protection from the drug. And by knowing what this network is, they identified another drug that can disrupt it, and that drug is pretomanid. And they showed that when you combine the two drugs, you have much more efficient killing of the pathogen. And these drugs, bedaquiline, pretomanid, and there's another drug, linezolid, which have been approved, and also delamanid, they have been approved for a multi-drug cocktail for treating TB. And this was the first cocktail to be approved in about 4 decades or so. So here's one idea. So these vulnerabilities that we're identifying is from our systems biology approach. What we're now doing is we're now collaborating with a company called Relay Therapeutics in Cambridge, Massachusetts, who have developed a technology called DNA encoded library screening, or DELS. These are essentially molecules that are typically found in the library of molecules that pharmaceutical companies use, but they are covalently linked to these DNA barcodes. So what we can do is we can take these vulnerabilities identified by our network approach, make proteins in the laboratory, send them to Relay, and they will then do DELS screening on those proteins to find molecules that are binding, and they can do it in high throughput because they can put all the molecules together and then just sequence the barcodes to know which molecules actually bound to the target. This information is then sent to Google Research. And our collaborator there, J.W. Feng, is applying machine learning and artificial intelligence approaches to the structures of these molecules to find patterns of structures and functional groups to come up with consensus structures that are likely to be effective. And we will then synthesize these molecules or purchase them if they're available off the shelf, and then do screening in the laboratory. So instead of doing brute force 150 million assays with 150 million compounds, we can now do quick machine learning based rational discovery of molecules that we can screen in the laboratory and get drug candidates. So this is one of the first ideas we're working with in partnership with these two companies. The other thing we have done is, as I mentioned, is finding drugs is one thing, whether they're working or not is a second important thing we need to do. For this, we want to replace bacteriological assays. So what Vivek did was he took a similar compendium of these gene expression profiles or transcriptome profiles of MTB from many different contexts. And then he trained a machine learning algorithm to find a signature in those data that could identify uniquely transcriptomes of the pathogen when it's alive. Okay. What you can do now is with this signature, you can give it a new transcriptome signature, which is very easy to get, much easier than doing a bacteriological assay. And you can ask the machine learning algorithm, is this pathogen alive or is it dying? If the transcriptome profile falls within this classified region, then the machine learning algorithm will say, yes, it is alive. If it falls outside it, depending on how far it falls, it will tell us, no, the pathogen is dying. So in that way, what we've shown is this is far more accurate than current bacteriological assays. So here's a study where the pathogen was grown in a flask and then transferred into a medium without potassium. And what that does is it pushes the pathogen into a dormant state. It goes into early, middle, and late stage of dormancy. At the same time, when we did this, when the group that did the study did the shift, they added isoniazid. And then they did colony forming unit counts, which is essentially counting how many bacteria are still alive. And based on that approach, the bacteriologic assay said that the pathogens are all dying. In fact, they're dying by 4 logs, which is almost as if you've cleared all the bacteria. But if you use another more sensitive method, which is difficult to do in a clinical setting, it's called the most probable number or MPN technique, and that can count both bacteria that are active and those that are dormant. You find that none of the bacteria were actually dying. They were just shifting into dormant state. They were still alive. So here it shows the bacteriologic assay is not sensitive. But if you use our approach of using this machine learning signature, we can find that the bacteria are actually all alive. This is the threshold below which the machine learning algorithm says the bacteria are dying, and the score never drops below that. So we then applied this to a clinical study where these patients were given a treatment regimen, and this is when they were given it on day 0. And then over time, the bacilli were collected from their sputum and then profiled for the transcriptomes. And the assessment of whether they were fully cured or not was done in 8 weeks. And that's the culture conversion, or it's a bacteriologic assay done in 8 weeks. But here, within about 2 weeks, we showed that this machine learning algorithm could identify patients where the cells were dying and patients where they were actually growing. So within 2 weeks, we can now give a report on treatment efficacy on a patient, which has immense value. But we want this to be a clinical test. So now we're working with partners at University of Cape Town and at Stanford University to develop this device, where what we're doing is taking advantage of some of the capabilities that were developed in University of Cape Town, specifically this instrument called respiratory aerosol sampling chamber that can capture bacilli from a normally breathing individual, asymptomatic individual, or we can also collect bacteria using the technology Eliza developed called PathSeq, which we can apply also to a mouse model. So the sample can come from a patient or a mouse model, depending on whether it's a human clinical trial or patient care, or whether it's a preclinical trial. Once we've captured the bacilli, they can be then channeled towards a microfluidics device, which is a small device that can be a few dollars and affordable for use in a remote location. And that technology is being developed at Stanford by Lars Steinmetz and his team. And they can perform what is called a loop-enabled amplification, or LAMP, or a linear amplification, which allows you to do amplification of the signature for the machine learning algorithm DRONA that we've developed for doing assessment. And this can be done at even a remote location. So we can take the signature and feed it towards this algorithm called DRONA. We've also shown that we can optimize LAMP to detect up to 1 single copy of the pathogen in a given sample. So we can do both, detect the pathogen and then send it to this algorithm, the machine learning algorithm DRONA, to do 2 things. One is it can quickly make an assessment to see whether that profile that we measured is from a living bacterium or a dying bacterium. And it can give a report on whether there's effective clearance of the pathogen. The second thing it can also do, and this is a future application and more in a clinical trial setting and drug discovery setting, is it can also predict combinations of drugs. And as I mentioned, drugs can interact, like the TB drugs and HIV drugs. So we want drugs that don't cancel each other's effects, but they promote each other's effects, or they're synergistic. And finding these combinations is a very difficult and time-consuming process, and currently done with what are called checkerboard assays or diamond assays. And with this approach, we could probably make it much faster and all in a computer, So we can then only select those combinations that are likely to be effective for doing these actual experiments. Okay. So I essentially told you what is needed to deal with the current TB pandemic, and two ideas that we've come up with to address all of these. The first idea will address the first three challenges that I described, and the second idea will address the remaining two challenges. And again, I mentioned people as I've gone along, but I just want to again list their names here because we couldn't do this on our own. So thank you. And if you have any questions, I'd be more than happy to answer them.
Thank you, Nitin. So we do have one question. Can you hear me?
Yeah.
Okay, we do have one question, and to start, this is one disease you don't want to get. How likely is someone to be exposed to catch it these days when traveling, especially given that Americans don't get vaccinated for it typically? I think that's question number one. Since it also affects the lungs, as early COVID-19 did, do we have an idea of whether having COVID makes people more vulnerable to this?
These are great questions. Regarding the first question, typically healthy individuals who have strong immune systems are not as at high risk for getting the disease, but that doesn't mean you won't get it. So it would be really important for you to take all the precautions and when you go to a TB endemic region where there's high rates of TB, make sure that your nutrition is good and you eat well. There are no vaccines right now, although there are some vaccines at late stages that are showing very promising results. There are some vaccines that are fairly effective at an earlier stage of your life, but they're not as effective. And this is slightly outside my expertise, but I do know that there are no effective vaccines for TB yet. As far as COVID-19 is concerned, there are some early reports that could increase the risk of getting TB, but it's way too early to tell. I think we have to wait to see, to have these studies completed and check to see if it increases your risk of getting TB. Two, if you have latent TB, if it increases your chances of getting reactivated. And three, whether it would essentially change the overall dynamic of how you would respond to treatment if you do get the disease.
Great, thanks, Nitin. Here's another question. Is there transmission from animals? I've heard of TB in elephants, for example.
That's a great question, and I don't know if there are any examples, and I don't know if that someone on the call from my team has better knowledge regarding this, of zoonosis of the pathogen. There are strains of the pathogen that can infect other species, but I don't know if those species can then jump over to the human host, and whether Mycobacterium tuberculosis can also infect other animals. I'm not 100% on that.
And I have a question for you, Nitin. You know, when we asked you to speak at a research roundtable, that you could have chosen many topics. There are a lot of things going on in your lab across a broad range of very interesting research. And you wanted to talk about this, which I made the assumption that you're feeling really good about it and positive, and it's very interesting to you. Can you say a little bit more about that?
Yeah, I think there's a few things. One is, I think I have a great team and great collaborators, and we're making some really good progress. It's taken us a long time to get where we are. And for me, it's very exciting to see that we're beginning to see some translation opportunities with different collaborators, and our work is being recognized worldwide as well. So I thought it was timely. I also thought it was timely because I think people are now more aware of what pandemic can do, and we've directly experienced the pandemic ourselves. Now I grew up in India, and I've had a different experience. I know people who got tuberculosis, some close friends, and so I had a different perspective. And but when I came to this country, I realized that some of those diseases are neglected, and I was very happy to see the Gates Foundation take great interest in supporting the research that no one else wants to support because it's not a money-making business. But I think all the stars are aligned. I'm not religious or superstitious, but I think things are aligned right now in terms of awareness, in terms of where the research is, in terms of technologies and tools that we can apply to the disease. And Seattle is an epicenter for TB research with a foundation here, but there's all these institutions, Seattle Children's, you have Bath, you have even at the Hutch, there's research going on. So there's a whole host of different, even the University of Washington, they have a fantastic global health department there, and we work with all of them. Everybody in the TB community knows each other here. And we've been recognized as an epicenter for TB research. So I felt, there's a lot of discussions around the COVID pandemic, and I just felt people should now kind of know that out of sight, out of mind, but this is something we should all care about. And frankly, with the way climate is changing, a lot of the tropical diseases, and also with travel, the world is flat. These diseases are not going to be restricted to these developing regions. They will travel worldwide. And we're seeing that happening already.
Great, thank you. And speaking of this, you're talking about Seattle being a hub for tuberculosis research. Did you know that at Town Hall Seattle tonight, the topic is tuberculosis?
Actually, I didn't know that. I should know that. Yeah.
Well, you know what? Here, I'm gonna surprise you with. I'm actually going to read a little bit from their synopsis of what they're speaking, and I want you to comment on it. And by the way, we love Town Hall Seattle. We're a fabulous partner with them, so we love telling other people about it too. This event is at 7:30 tonight, and it is. Let's see who's speaking. It's Vidya Krishnan and Amber Payne, the past, present, and future of tuberculosis. According to the World Health Organization, over 1.5 million people died from TB in 2020. Could it be on track to reemerge as the next global public health crisis? Your thoughts, Nitin?
Yeah, Bill Gates had predicted that there's going to be on the verge of a major pandemic, and COVID is a major pandemic. But the bigger concern is a bigger pandemic might be on the horizon, especially with drug-resistant strains for different diseases cropping up. So, I don't want to believe on a down note. There has been great progress on TB, and the rates of TB and the fatalities because of TB have dropped significantly. There is concern because there's multi-drug and extremely drug-resistant strains that are coming up. So all the progress that has been made so far could be for naught if we don't really act post haste to deal with these new resistant strains of TB. And so we need ways of monitoring with accurate tools the transmission of TB, even in asymptomatic individuals in high-risk regions. And so a lot of these tools don't exist, and that's what we're trying to do. And then second, if you do detect transmission, if you do find patients with disease, we need effective drugs. So it's a systems problem. So I think I'm glad to hear that they have a conversation. Maybe I'll attend it this today.
Yeah, and one of them just published a book too, so you can get the book as well. Okay, here's our next question. Do we know what percentage of patients who start treatment actually complete it? Presumably it's a fairly low number and depends on accessibility.
I don't know the exact numbers, but because of the burden of the treatment regimen in terms of how many pills and for how long and all the side effects and so on, and not knowing whether it's working and so on, a lot of patients do stop taking their drugs. And the lack of compliance is believed to be one of the major reasons why drug resistance becomes a problem. And percentage-wise, I don't know how many people exactly stop taking the treatment, but there are efforts like the DOTS effort, which is part of the End TB campaign by the World Health Organization, where it's directly observed treatments, which is a clinician actually observing a patient taking the drugs and making sure that they are compliant. And that actually improved substantially the treatment or cure rates and so on. So that is a big issue. And that's one of the reasons why those challenges are important to address. We need pills that are effective in killing the pathogen faster without having all the side effects.
Okay, great. Thank you. Next question. Will the mRNA technology vaccines be applicable to TB? Has the COVID pandemic and the science done around it helped to advance any of the issues present with TB?
There's definitely been discussion about the mRNA vaccines are it's such cool technology. I mean, you can program it very quickly and you can program it to express the antigen of any pathogen. The challenge will be in terms of whether the immune system is effective enough against TB in the immune compromised setting of many of these individuals. So there might be a combination of one, having the vaccine, but also having some ways of kind of stimulating the immune system so that it is more effective in clearing the pathogen. But there definitely are talks about applying the technology to TB and a number of other diseases.
Okay, great. So this question, the person who's asking the question is noting that you have already said that you are not superstitious, but would you look into your crystal ball? What are your predictions about how the TB pandemic will look in 5 years, 10 years? Is it solvable?
It's there's a big debate about, you have 2 billion people latently infected with TB. Unless we find a way of, look, the thing is to first make sure we are able to treat people who have active disease, the stop transmission. And then you have to also deal with the latent TB infection. The big debate is, it's very difficult to kill TB in its latent form. People who are not symptomatic, doesn't make sense to give them very toxic drugs, one. Number two, there's this wake and whack idea that's been discussed, which sounds very cool, where there are ways you could potentially trigger the pathogen to replicate. So it kind of reveals itself and then you whack it with drugs, right? But there's big ethical issues about whether that would actually exacerbate the situation. We don't know what the clearance rate might be. And it's a big debate there. So because of the large scale latent infection of by the pathogen, 5 to 10 years, we won't see it solved. We might see some good progress in terms of vaccines. So it could be that you will see protection against new infections.
Right.
You will have new drugs that will be more effective in treating TB, hopefully also for multi-drug resistant TB. And in fact, drugs that are more effective in treating to complete clearance so that you don't have resistant strains emerging. So I think you'll see a lot of really exciting progress in the next 5 or 10 years.
Great. That's good. And this is your last question. What do you believe is the next biggest technological advancement that will help fight TB?
I want to say the devices we're developing, but there's a lot of advancement in immuno-oncology and cancers, for example, immune therapies. I think the granuloma in many ways is kind of like a solid tumor with an immune suppressed environment with exhausted T cells and everything. And there could be some major breakthroughs that will hit on a lot of these other challenges I've touched on in terms of activating our own immune systems to be able to go and clear. So if that is possible, then, and if we can detect some of the incipient TB, so we have monitoring devices that can detect TB in asymptomatic individuals, even before you see, through radiology that there are any abnormalities, so before it's caused any damage, then you will have a way of, early detection is always the best, then you will be able to clear it much sooner. So there might be some advance in vaccines that I think would be major breakthroughs in activating our immune system, similar to how immunotherapy works. And third, in effective monitoring of patients.
Okay, great. Well, thank you, Nitin.
Sure.
Yeah, thanks for joining us today. And I know we'll have you back, and we are working with your team too to get a STEM education research roundtable going, because we have a great story to tell there, and we're always learning more about that all the time. So thank you very much. And we have a couple of events coming up for our guests. Thank you so much. Town Hall Seattle, again, our friends there tonight. That's at 7:30 tonight. And that is the past, present, and future of tuberculosis. And that'll be a really interesting way to kind of continue the discussion of what we were just talking about. You'll have to buy a ticket at Town Hall Seattle. Those are $5 each. And you can watch that from the comfort of your home. Our next research roundtable, we have a date. It's in May. It's May 12th, and it's Samantha Picos and it's Jen Hadlock. And they'll be talking about recently published papers and materials about COVID and pregnancy. And so you'll definitely want to tune in for that. And we're working on getting some research roundtables, one for March and one for April too. So we'll keep you updated on, you should all be hopefully on our email list, and we're easy to find. So thank you very much for joining us tonight, and have a lovely evening. Good night.
Good night.